A very happy morning to everyone. Today we are going to discuss about C3 cycle. The C3 cycle is also known as Calvin cycle. Melvin Calvin and his associates Andrew Benson and James Basham discovered this pathway by using chlorella and Saint Dismas and hence named this as Calvin cycle. Calvin cycle is also called photosynthetic carbon reduction cycle, which is PCR cycle, or reductive pentose phosphate pathway, which is RPP cycle. The Calvin cycle includes three steps, that is carboxylation, reduction and regeneration. Let us study about each step in detail. The first step is carboxylation. In this phase, 6 molecules of carbon dioxide are accepted from the atmosphere by 6 molecules of the primary carbon dioxide acceptor, that is ribulose 1,5-bisphosphate, which is a 5-carbon compound to form 12 molecules of 3-phosphoglyceric acid in presence of water. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase or Rubisco enzyme. The enzyme Rubisco is present in large amounts in stroma of chloroplasts. The second step of the Calvin cycle is reduction. These are a series of reactions that lead to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per carbon dioxide molecule fixed. The reaction takes place in 2 stages, the first using ATP produced and the second using NADPH produced in these reactions. The overall effect is to reduce a carboxylic acid group to an aldehyde group. The product is a 3-carbon sugar phosphate, that is 3-glyceraldehyde phosphate. The third step is regeneration. Regeneration of the carbon dioxide acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP consumed in the first reaction. This process involves a complex cycle containing 3, 4, 5, 6 and 7 C sugar phosphates. For every carbon dioxide molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. This pathway is also known as C3 cycle because the first stable product in this cycle is a 3-carbon compound, that is 3-phosphoglyceric acid. For reducing 6 molecules of carbon dioxide into an hexose sugar, a total of 12 molecules of NADPH and 18 molecules of ATP are required. In summary, 6 carbon dioxide molecules will be taken to form 1 glucose and 18 ATP will be taken to release 18 ADP and 12 NADPH will be taken to release 12 NADP. For the net gain of 1 triose molecule, 9 ATP, 6 NADPH2 and 3 Calvin cycles are required. For the formation of 1 hexose, 18 ATP, 12 NADPH2 and 6 Calvin cycles are required. For the formation of 1 sucrose molecule, 36 ATP, 24 NADPH2 and 12 Calvin cycles are required. And some of the properties and functions of Rubisco enzyme. The Rubisco enzyme is the most abundant protein on earth. It constitutes 16% of chloroplastic protein. It is synthesized by both nuclear and chloroplastic genes and bifunctional nature, and it shows both carboxylase and oxygenase activity. Affinity for carbon dioxide is greater than oxygen. Carbon dioxide and oxygen are competitive inhibitors for active site. Affinity for oxygen increase in high light, high temperature, age of plant and oxygen concentration. And that's all about the Calvin cycle. Thank you.